And it is my distinct pleasure to today introduce Roshan Rao, who's going to be talking about EvoScale and ESM3. Just briefly, he previously worked at Meta AI and also completed his PhD at UC Berkeley, where he developed methods for protein structure prediction, function prediction, and design. Now at EvoScale, he primarily is focusing on the development and understanding of foundation models for proteins. So without further ado, Roshan, please take it away. We're very excited.
All right, thank you so much for having me. Yeah, I'm super excited to present our work on ESM3, which has been in progress for all a year now. Evolutionary scale was started officially almost exactly a year ago today. Yeah, this is work that has been joint among the entire company. You can see by the author list. So I'm presenting this, but none of this could have happened without all of the people on this list. So overall, I'm going to be about ESM3, the architecture, the training, and the way we use and sample from the model. Go into how we can prompt ESM3 to design new sequences and structures. Talk a little bit about aligning ESM3 to produce structured sequences with certain properties that we think are desirable, and then talk about our validation in the form of generating a new green fluorescent protein, and finally discuss some next steps for the company. So ESM3. I think an overarching goal for ESM3 is really to take a really big step past where we were in ESM2, and see what we can learn from it. And I think that this was a motivating factor because we really thought that the space of protein models has been in a bit of a local optimum where people know what works well, reasonably well, and are able to like train new models in that space. But we think that it's possible to get to a different level of performance and to really have models that are capable of much, much more than existing models. And so ESM3 was designed to just really move as far as we can in a direction that we thought was interesting. And I think what we want to do now is understand what it's capable of, what new abilities it has unlocked that are beyond ESM2, and then whether any of the decisions we made were not optimal and whether we can improve them for the next model. So in particular, there are a couple of core ideas behind ESM3. The first one is one that's been behind this team for many years, and that's the idea that scaling compute parameters and data can lead to better representations and that better capture the underlying biology. So one example of this for ESM3 is that we really sought to scale things like metagenomic data. And we've actually explored metagenomic data for many, many years and have generally found that the model performance decreases when you add metagenomic data. When we finally started looking at this for ESM3 again, we realized that there's actually a crossover point where once you have enough parameters and enough compute in your model, that the introduction of metagenomic data starts to increase the performance of these models. So overall, this is like a really key part of ESM3 and of the philosophy of the team as a whole. We also really wanted to look at going beyond sequence data, and there are a couple of reasons for this. One, we thought that it would learn representations that were more informative, that were more tailored to what people tend to want to specify when designing proteins, when predicting structures, and would improve the performance in several downstream tasks. But beyond that, I think that language models really are very powerful, but they learn exactly what you tell them to do during the training process. And so when we train on sequences, they learn the distribution of sequences, but they don't make any special effort towards learning other characteristics. Anything else that they learn is sort of this purely happenstance, where if it's helpful for learning the statistics of the sequences, then the representations will capture this underlying information. But we've seen in places like natural language processing that if you simply train on it's better to train on the set of all the things that you want the model to be capable of, and then to fine tune or to just prompt the model to do the task that you're particularly interested in. And so incorporating this data, we thought, would steer the model towards learning representations that were particularly useful for tasks that were important, that had structure or functional components. The third point that we were looking at is the idea of synthetic data. And synthetic data has been explored in these models for some time. AlphaFold 2 first used a large amount of synthetic structure data to do self-distillation. But ESM3 takes it to a very different level, training on a very, very large set of synthetic data across multiple modalities. The other thing, when we thought about creating these different data modalities, it's possible to design many, many architectures here. And one thing that has been brought up is that you could design independent models on sequence, on structure and function, and then fuse these towards the end. But we thought, like, as you start to train very, very large language models that have billions of parameters, these models are capable of ingesting a very high degree of information and processing it. And so it's kind of a waste to keep this information separate and only allow the model to fuse it at the end. Whereas if we're able to inject this information at the very beginning, then the model can simply sort out what it wants to learn from each of the modalities. And then finally, something that was very important as we thought about how are we going to scale this model? How are we going to make sure that it trains stably and that it has high efficiency and that we know what's going to happen as we scale to 10 billion or 100 billion parameters. It was very important that we have a simple and scalable architecture and training objective. And we thought that by enforcing this and having a simple transformer architecture and a simple language modeling objective, that this would scale very well and that this was actually powerful and expressive enough to have strong generation capabilities and strong representation learning capabilities. In particular, we achieved this simplicity and the scalability by enforcing tokenization on all of our modalities. So this is the architecture of ESM3. There is a set of input tracks covering sequence, structure, and function, and a set of output tracks covering the same modalities that are passed in as input, which creates a very symmetric input and output to the model. The overall architecture is just a series of transformer blocks with nothing particularly significant in terms of architecture, with the exception of the very first transformer block, which also gets in the exact 3D coordinates of any structure that we want to specify. And this is important for a variety of reasons. One of the key things that we found is that the performance of things like inverse folding and anything that requires structure to be provided as a prompt improves significantly when you provide the actual coordinates as input. And it's unclear if a better tokenization scheme could help us avoid this, but just empirically we found that this was actually very important for achieving the best performance when generating sequences conditioned on structure. So we've seen generative models of proteins in the past, and a lot of them tend to be diffusion models. What we're looking at here is a model that is more like a mass language model, and it's based on this paper from Cheng et al. Called MassKit. There's also some connections with discrete denoising diffusion models that have come out more recently. But this is essentially what's happening with ESM3, is that ESM3 is going to start from a masked set of tokens, and then it is going to iteratively decode the structure of, or the sequence or structure of a protein based on some initial set of tokens. So this, for example, shows a generation where ESM3 was given the residue coordinates and amino acid identities of three residues, and as well as a functional prompt, alpha beta hydrolase, and then it generated the sequence of a protein. And what we're showing over this process is the coordinates that correspond to the tokens that were unmasked at each step. So this goes from a fully masked or just the prompt to the coordinates at each step. And we can show an example of this being built up in real time, where the model is given these key amino acids as the prompt, and it decodes the remaining structure. There we go. So we actually looked carefully at doing this type of learning in directly in coordinate space. So passing the coordinates into the model, having the model produce coordinates at the output. And this of course works. Like you can train a model to produce 3D structure directly at the output and backpropagate through it directly. But it does have some problems. It's very memory intensive. It has very different gradients than a standard mass language modeling loss. And it's actually computationally quite expensive. If you look at a standard AlphaFold 3D coordinate loss is quadratic in the length of the sequence. And so we wanted to try and move away from having to do this when training very large models, because we wanted to ensure that they would train stably, scalably, and would not lead to any type of divergence like very late in training. I think there's a question. How do we decide which are the key components versus non-key components when providing input prompt ESM3? I think that's it's really depends on what you want. So how do we decide what are the key components when providing a prompt to ESM3? In this case, the prompt is, this is designed to be illustrative, so we wanted to prompt with the sequence, partial structure, and partial function. But in general, it depends on like what is your goal. In the same way that you might ask like, okay, I want to generate an image. What do I want that image to contain? We want that experience to be very similar when using ESM3. So like, do you care about maintaining the overall fold while changing certain details? Do you care about maintaining the the positions of a key active site, but you would like to change the fold entirely? So depending on what you're actually interested in having the model generate is what you should specify. And overall, the model is relatively faithful to the prompt. It can be aligned to be more faithful to the prompt. Yeah, one of the failure cases we actually find is when you specify something that's like physically impossible. So if you specify a set of coordinates that just simply cannot be realized, then the model doesn't fails to generate it, and that's something that has to be worked out. Is to make sure that whatever you're prompting is like physically realizable. Another question is, could you share more on the optimization issues you met when not using structural tokenizer? So it's actually interesting. We did a lot of work on stability and on optimization to make sure things trained very stably. And ultimately, we had very few optimization issues. However, this was all at the like 1.4 billion parameter scale, and there's going to be different optimization issues at the 100 billion parameter scale. And there were parts of it at the 1.4 billion parameter scale that kind of raised some red flags and made us worry about scaling to 100 billion parameters, where we thought, okay, if we were to scale this to 100 billion parameters and it failed halfway through training, that would be a very expensive mistake. And some of those parameters were things like, or some of those red flags were the amount of memory usage during the computation of losses for coordinate-based losses, the relative scale of the gradients of coordinate-based losses versus mass language modeling losses. And the speed of computation too. We just saw that the efficiency takes a big hit when you are directly predicting in coordinate space versus when you're predicting in token space. So those were some things that we thought could maybe work at a smaller model like 1.4 billion scale, but at 100 billion parameters, we're very worried that those would become a big issue. I'll talk a little bit, I'll show some training curves also a little bit later into this talk, and talk a little bit about some of the details that are involved in making sure that these models train stably. So we looked, so when we were trying to tokenize these when we were trying to ensure that everything that's input and output from the model is in the form of tokens, one of the key questions is like, how do we tokenize structure? And there's been some work on tokenizing structure with fold-seek, but we really wanted to make sure that the representation that we used was able to reconstruct the structure of the protein with very high resolution. And we were worried that the tokens from fold-seek are not high enough resolution to actually reconstruct the full 3D structure. So when we were training ESM3, we trained a new VQVAE, which takes the backbone coordinates, produces a token for each residue in the protein, quantizes it, and then a decoder is learned to predict the structure from these quantized tokens. A key thing here is that each residue actually represents the 16 nearest neighborhood around the around each residue. So residue I represents the 16 nearest neighbors in 3D space of residue I. So the tokens are actually local and contain local structural information rather than global structural information. There's a architectural piece here that is incorporated both in the encoder of 3D structure as well as in the first layer of ESM3 when we're taking in 3D structure. And that is this concept of geometric attention, which is a architectural innovation that is designed to encode the 3D structure of a molecule in a way that is fully invariant to SE3 rotations and transformations, but which is also very, very efficient to compute. So the state is it's designed essentially on the attention operation, where the state is split into queries and keys, and then attention is computed rather than on a dot product, but based on the distance between predicted queries and keys, as well as the dot product between, or like the cosine dot product between the normalized dot product between the queries and keys. And the reason we split the attention like this is that this should fully specify the positions, or allow the model to learn how to fully specify the position of each amino acid relative to the other amino acids in the context. But because it's decomposed into these attention-like operations, we can actually write a very efficient CUDA kernel that's based on the flash attention kernel to compute this operation and have it run extremely quickly during training and during inference. So this ends up being not quite as fast as standard attention, but only like about half to a third of the speed of standard attention of an optimized flash attention kernel. And here's a visualization of some of the latents that the structure encoder learns. So this is essentially showing what specific tokens in the structure and the structure encoder learn to represent. So for example, we have a token that represents these parallel data sheets. We have tokens that represent this sort of alpha helix, adjacent alpha helix construction. These are some examples of tokens that represent relatively similar sets of structures. We do find there are some tokens that end up representing things that are fairly different from each other structurally. And so there's like a lack of monosemanticity in the tokens. That's something that we want to work on and to make sure that the tokens that we learn are relatively monosemantic. So that's something we'll hope to improve, but many of the tokens that are learned do have some sort of clear structure to what they represent. And again, our goal was to produce a structures or tokenized representation that allowed for very, very accurate reconstruction of 3D structures. And we find that this is essentially true of this tokenization, which generally has sub-angstrom RMSD when encoding and decoding a structure. And so this is not. So we are able to make predictions in structure token space and know that if they're correct, they can be decoded into structures with very high accuracy. There's some examples in the supplement also of cases where the RMSD is much higher. And this tends to be when you have long unstructured loop regions where there aren't really any contacts between, say, two domains that are connected by a long unstructured loop region. And this makes sense because the tokenizer represents the local structure. And so it's not able to precisely position the two domains. But I mean, in practice, those structures are either like unstable or they're not. They're only stabilized by the presence of some other protein that is not in the tokenizer's context, like another multimer. And so it's reasonable for the tokenizer to not have high or low RMSD on those types of structures. Okay, what did we do to scale ESM3? So these are the hyperparameters for the three models that we trained out, 1.4 billion, a 7.7 billion, and a 98.5 billion parameter model. There are a couple of interesting components here. So the big things that are scaled are the number of layers, the model dimension, and the number of tokens that are trained on. So starting with the number of tokens, this is based on chinchilla optimal scaling laws, essentially, where there is some theory around how many tokens a model needs to see in order to be compute optimal. Where if you were to train for more tokens, it would have been better to train a larger model instead for the same amount of compute. There's also the relationship between the number of layers and the model dimension. where for smaller models, or for models in natural language processing, you typically see that models scale in the model dimension much more so than they scale in the number of layers. And we found that it's actually better to scale in the number of layers for proteins rather than the model dimension. And we found this is particularly useful for predicting structure. And there's some theory behind this. I think that one piece of information is that like smaller vocab sizes, where in amino acids there are only 20 distinct tokens, generally tend to perform better when you have deeper models and you don't need as much as many parameters in each to represent each amino acid. This is analogous to in vision, you also tend to need deeper networks rather than wider networks. And also, like we've seen this in other models that have been successful for proteins. AlphaFold is relatively deep for the number of parameters it has, and recycling essentially acts to increase the depth of the model without increasing the number of parameters. So it seems like proteins like deeper networks than shallower networks. This is the data that we trained on. Like I said, that there's like a pretty large set of metagenomic and synthetic data that was used in the training process. And here is some training curves. So I think this is something that's not in the paper, but this is showing the training curves for, and the green line is a 98 billion parameter model. This gray line is 1.4 billion, and then the maroon and blue lines are the 7 billion parameter model. And you can see some of the metrics that we were monitoring over the course of training. And there's a lot of interesting things here. I'll touch on a few key things that were important for efficiency and scalability. So for efficiency, we were training not in half precision, but actually in FP8 precision. Some operations are optimized using transformer engine. The model is sharded over 1,000 or more GPUs. The activation checkpointing is important for reducing memory usage. Bin packing is very important for making sure that every single batch has maximum efficiency and you're not wasting a lot of compute with pad tokens in each batch. And so these are very important for efficiency. And then for stability, applying layer norm to the queries and keys, having longer warm-ups and scaling the learning rate depending on the model size, as well as ensuring that there is FP32 for some key operations, and having and experimenting a little bit with the masking schedule was important for maintaining stability of these models during the training. How do you interpret the jumps and peaks in loss over training? So there is actually a fair amount of literature about this when training large language models. So there are a couple of different jumps and that matter differently. So when you're training large language models, you expect to see these very large spikes in the loss. They are very common and they appear in whether you're training on text or images, etc. And what you want to see when you see them is that the model's loss goes, that the model recovers back to the loss value that it would have had if it didn't have that spike. So you can see that this, roughly speaking, happens over the course of training. And that's pretty important. Otherwise, you're going to see, worse performance than you would expect. The kind of loss that you want to, you really want to avoid losses where the model does not recover back to like where it would have been if the loss spike didn't happen. That indicates that you have a serious issue with the training stability. There are other things, like for example, there's a spike here, and this is not due to the model like failing or a stability issue, but rather this is because we switched the context length and a couple of other key things from 512, which is what this is trained on, to a higher context length and make some other changes to the data mixture in the last like couple percentage points of training, and that's just to improve the performance at the end. So yes, it looks like there's a spike here, but that's because the underlying data mixture changes there. How important do you think the SE3 equivariant attention is for the performance at this scale of data? What drove that decision? I don't know, honestly. I think that you could probably achieve similar performance using data augmentation or some other way of incorporating it. We found that this worked reasonably well and was very efficient to incorporate into the model. So it wasn't really something that we didn't really look at non-SE3 models, although I think it's worth taking a look and seeing if there's any performance. Might have missed, but were disordered proteins regions used in training in any kind of way? I mean, the model is trained on basically every sequence in all of these data sets, with the exception of, I think, OAS. There's some sequences that aren't included just because OAS is very large. We didn't want to bias the model towards antibodies too much. So, I mean, as far as disordered proteins existing in Magnify, UniRef, and JGI, like certainly they are included in parts of training. And if there are functional tags in like interpro that are associated with disorder, they are included in training. I don't think we specifically looked at disordered proteins and regions as like a way of evaluating the model, but they are certainly trained on any protein that's in these data sets and on any functional annotations that interpro picks up for those data sets. I'm going to move a little bit faster because there's a reasonable amount of this slides left to go. But we see generally that as we scale the model, the loss decreases consistently, as well as the loss conditioned on other modalities decreases consistently. And this so this is essentially what we would expect to see. And cool, so let's shift to talking about some of the results that we got from prompting ESM3. There's one more question. Why were the backbone direction and distance losses clamped? The distance and direction losses are clamped just, it's if you look at the values are based on Euclidean distance, and so they can be very, very high. And so essentially we clamp them to avoid very high gradient values that would cause instabilities. Although those losses are only used when training the structure token encoder rather than ESM3. Cool. So the first thing we looked at when generating proteins from ESM3 were some basic generative capabilities. So that's like freely generating proteins from the model. And this results in a set of proteins here that mimics the distribution of natural proteins. So the gray is essentially the distribution of natural proteins in the PDB, and the colored points are samples from the model that mostly fall within this natural distribution. You can see a different view on this here, where gray is the PDB background, and this is the distribution in alpha and beta of ESM3. You can see that there's some proteins that have high proportion of beta, some high proportion of alpha, and a good portion that's mixed. I think a key thing when you're looking at these models is to not expect them to go beyond the training distribution unless they're specifically told to. So what we wanted to do is show that if you prompted it carefully, you could actually make the model generate proteins that are outside of the training distribution. So this is using secondary structure and SASA to prompt ESM3 to generate a protein. And you can see that while unconditioned generations have very high similarity to the training distribution by default. The condition generations have much lower similarity to the training distribution by default and much lower sequence identity, both structural and sequence similarity. You can also do the same thing on symmetric proteins, prompting the model to generate proteins that have symmetry. And this is essentially done by just having a symmetric prompt in the secondary structures. Another thing we wanted to look at is the composability of prompts, because there are many ways of providing a prompt to ESM3. And so we really wanted to look at whether we can ask the model to satisfy two different constraints at the same time, or multiple different constraints. So this could be something like asking it to maintain the structure of this motif while having this fold, which we see it's able to do very well. And we were able to show this across a variety of different combinations of prompts, where the model maintains both high ability to correspond to the fold or the secondary structure that's asked for, while also maintaining the motif that is generally requested. Another thing we looked at is compressing a structure, so maintaining the overall fold and ensuring that this particular catalytic region was maintained, but decreasing the length of the protein significantly. Was the compressed serine protease trypsin domain protein expressed or well tested? No, we did not. So essentially, the GFP is the wet lab prediction here, or the wet lab validation here. We're very interested in doing generations of this, but the goal of this figure was to show like computationally what's possible under this model. But of course, like we're not arguing that these are necessarily real proteins until someone actually makes them in the lab. Did I miss a question? Why does it seem like adding function tokens loss does not improve much? Yeah, this is an interesting question. It's something we want to look into more and see if we can have a better representation of function in the model. I will say the loss improves the most when so the way we computed this is to do like a decoding order where we do like iterative decoding through the model. And function tends to decrease the loss the most early on in that process. But later on in the process, like once the model is decoded 85% of the sequence, the function doesn't really add much new information. So that's one potential reason why the function is not as informative. But I think it's something that we're trying to look into. To evaluate the model for inverse folding tasks, how does it compare to other tools, protein MPNN, et cetera? We have, I mean, this is the loss of the sequence conditioned on the structure. So you can compare that to what protein MPNN or ESM-IF1 or another model achieves for structure conditioned sequence perplexity. Or this is the loss rather than perplexity. I think that, yeah, so I think that's what we did. It's kind of hard to compare, I think, in terms of like if you want a comparison in the lab, then obviously you need to generate actual lab tokens, actual lab results for these. There are a lot of questions. I might have missed some. How is function tokenized? There's a single token per function. There's a lot of details in the paper on it. It's basically tokenizing the keywords that are associated with interpro annotations. And so that's what we're doing, but there's details on exactly how that happens in the appendix. So it's a little too technical, I think, to go into right now. Cool. Okay, let's talk about alignment. I think alignment is something that we're really excited about. I think it's something that we think will be the future of protein language models, and will have a lot of impact on how they're used and what actually makes them useful. So what is the idea behind alignment? Well, the idea behind alignment is that language models are very powerful and they have a latent representation of the space that is really strong, but they're trained to fill in the blank essentially. And so they're trained to reproduce their natural distribution of proteins, and that may not really be what you want. For example, like the natural distribution of proteins has many proteins that uh, maybe don't fold particularly well in the lab, that aren't predicted with like high PTM, and that which and kind of longer term maybe proteins that are not very stable, that are not soluble, that and you might want instead to make sure that the model will very consistently produce proteins that have high PTM, have low CRMSD to the prompt, proteins that in the future might be very stable when expressed in the lab and will fold with high accuracy. And so alignment in theory can help us accomplish some of this. So the way alignment works is that you take a prompt, you generate a bunch of sequences from it, you apply a scoring function to the generated sequences. In this case, this is purely computational, so there's no lab involved. And so we're just scoring the generations based on the constraint site RMSD, CRMSD, where we look at the prompt and the predicted structure of the generated sequence, and we say, does this predicted structure match the prompt at the key residues that were specified, as well as whether the predicted TM score of the structure is high or low. And then in fine tuning, the model is trained to maximize the log likelihood ratio of the high scoring generation over the low scoring generation. So this is the overall process of alignment. We see that we evaluate this on a prompt following task where we ask the model to generate a sequence which has a structure that will fold into something that has a certain orientation or certain position of key residues in space. So something like this, where you would give like the 3D coordinates of these positions and the amino acids of them and say, okay, give me a sequence that will fold into a structure that has this motif. And you see that with as model scale increases, the performance of this, like it past that 128, is how likely the model is to generate at least one successful protein given 128 samples. And we're showing this over a variety of different prompts. So like with if it's 1, it means that it generated at least one success for every single prompt. At 50%, it means that for 50% of prompts, it generated at least one success. So you see that like there is an increase in performance with model scale. But after fine tuning, we see that all the models get much, much better at following the prompt and generating high quality sequences. But that in particular, larger models respond more significantly to this alignment process. And there is that after fine tuning, the 98 billion parameter model very consistently generates proteins that have a high fidelity, follow the prompt well and have high PTM. You can see that in general, this seems to increase the diversity of the generations as well. So this is showing the number of structural clusters, or the average number of structural clusters for successful generations. So after fine tuning, the model is generating more successes and more diverse successes. And that the overall distribution, so if we look at it on a per prompt basis, where each kind of ligand annotation here is describing a prompt. There's a table in the supplement that tells you exactly what the prompt is for each of these. But it's showing like the distribution of generations and how it shifts between the base model, which generally has like relatively high constraint site RMSD and relatively low predicted TM score, and the fine-tuned model, which usually has like much, much lower RMSD and much, much higher PTM. Can you comment on the choice of the IRPO objective for alignment and whether you explored any other preference optimization objectives for alignment beyond DPO? We looked at IRPO versus DPO. We found empirically it seemed to improve performance somewhat, and it was relatively easy to incorporate, so we chose IRPO. We did not particularly look into other methods for alignment beyond DPO. So things like online RLHF or other methods we didn't actually look at. DPO is nice in that it's mostly offline and relatively simple to implement. So we chose to use DPO. Surprising to you the 1.4 billion model performs so poorly. So this is interesting. This task is chosen to be deliberately very difficult, because we wanted to really get at very difficult tasks where you would see like very, very large changes in model performance. And so so in this case, like there are a lot of details in the dataset construction that are really asking the model to generate something that is very, very different from its training distribution without a whole lot of information. And so it is true that the 1.4 billion parameter model does relatively poorly, but this task is actually constructed in general to be relatively difficult. And I don't actually know numbers for RF diffusion off the top of my head, but I think RF diffusion performs about on par with our 1.4 billion parameter model here, but I would have to rerun it. There's been some changes since we made this data set, and so I'm not 100% sure about that. Okay. Cool. Let's talk about the wet lab validation that we were able to do, which was focused around GFP. And we chose GFP for a couple of reasons. Like one, like fundamentally GFP is nice because it's very easy to evaluate. We can, we wanted, like we were training this model and we wanted to be able to evaluate whether it was useful in a lab, but we're fundamentally a computational AI company and didn't have a lab, especially at the time. So we wanted to make sure that whatever we chose to validate was relatively straightforward to do so. But beyond that, we thought that GFP was actually a very difficult mechanism to actually go after because the underlying physical mechanism for GFP is very unique. You have the way GFP actually functions is that you have this alpha helix configuration, which is in the stable pre-reaction configuration state of GFP. The side chains of particular amino acids will start to coordinate, and then they form this chromophore, which results in the breaking of a number of these bonds and causes or allows the molecule to absorb and then re-emit light. And so this is a very, very interesting and unique mechanism and requires a very high degree of physical of structural accuracy in order to form this exact site. So we thought that this would be a easy to validate but challenging design problem. So GFP ESM3 is prompted by providing the structure of the prompt and as well as or the key chromophore, and as well as the identities of some of the residues. And the model generates sequences based on that. There's an algorithm which we talk about more in the supplement, where the sequence and structure of the model is are jointly optimized. And this results in some really, so basically you want to make sure that the sequence has high probability under the model given the structure, but that the structure also has high probability under the model given the sequence. And this seems to result in higher success in the wet lab. It's a quick video of GFP being generated. And so this is showing the actual decoding order of the model for the molecule that actually glowed in the lab. And to touch briefly on like the success rate and the process that was used, we took the original GFP structure, extracted that prompted region from it, generated a series of sequences based on that algorithm that was shown, and then did a one plate of experiments where we found, in particular, there is like one example here that had faint activity that was around 57% similar to the wild type. Then we took this one protein and did some diversification and further refined it, and eventually found a set of proteins that have high activity, but one in particular that had very high activity and activity similar to wild type GFPs, and which has it's kind of identity. The sequence identity is relatively different from other GFPs. So we're placing the designed GFP on a plot here with natural GFPs colored various different colors around it. And here's a visualization of the mutations. I think something we're interested in is a number of mutations that are actually on the core and inside even the alpha helix of the GFP. So we thought that it was interesting that we were able to mutate residues that were in seemingly relatively important positions. Some questions here. In the paper, you explained fine-tuning, but could not find an example in your GitHub repo. Steps needed for fine-tuning the model with a new data set. We talked about, in the paper, talks primarily about DPO fine-tuning, or I think in the supplement there's some other fine-tuning. I mean, I don't think that there's any particularly special fine-tuning code. Like I would say, we've applied like LoRa. There's a repository called MinLoRa on GitHub, which works pretty well for applying LoRa to arbitrary transformer models. And I think that we've used that internally to fine-tune ESM3. DPO is more complicated. I don't think we were planning on releasing an example of it, but there are examples of DPO out there that I think you could adapt for ESM3 relatively easily. So yeah. Pranam asks, how did you choose the 3 model sizes and what happened to all the intermediate parameter size models? Well, okay, the 3 model sizes are chosen. We just did a lot of work at the 1.4 and 7 billion parameter scales. We thought those were pretty good for initial experiments and for showing the promise of these models. And then the 98 billion scale is simply the model that we had the capacity to train, given the time that we had compute for. And I think that the intermediate model scales are, if someone wants to give us more free compute to spend on training intermediate models, then I guess we'll train them. But I think that we just wanted to go and try and understand really like what happens. Is there a significant difference between models that are at the 1 to 10 billion parameter scale and models at the 100 billion parameter scale? So I think that's a scientific question that we're trying to look at and trying to answer now. Restriction on viral proteins will in fact aid safety efforts versus inhibiting efforts. The viral question is interesting. I mean, there's a lot of questions right now in the, or there's a lot of worry right now in the community around biosafety and the utility of these models for designing viruses. I think that the viruses were removed from the open model out of an abundance of caution. I think that that's a decision that we came to after talking to a number of people in the biosafety community who expressed concerns. I wonder if we'll be able to revisit that in the future. And we also hope that like the internal models do not have viruses removed, and so we hope that we'll be able to partner with academic labs and provide them access to those models if they are interested in pursuing research on viruses and vaccines. Would it be possible to prompt to generate models that minimize sequence identity to wild type? Yeah, so we don't, that's not a prompt that we support natively, but it should be relatively easy to fine-tune models, fine-tune ESM3 to generate sequences using the same methods that we use for the DPO. You'd simply have to change the score function to be based on the distance to wild type rather than anything else. On data, did you observe any variance in the value of data across modalities? Is structure function data more valuable than sequence? How do you handle deduplication quality control? The value of data is a really interesting question. Like the highest value data is PDB data. Like very clearly, I think that if we had 10 times more PDB data, that would be like would make models even like much better. But beyond that, I think that, it's possible that if you were to choose between one sequence and one structure, you might prefer one structure, but that we actually see that because there's so many sequences and the collection mechanism for sequences is so unbiased, or relatively unbiased relative to structure or other annotations, that the models learn really well on sequence, and that the model loss on sequence is very linear with scale. And so models like just continue to get better, even at the 100 billion parameter model scale on sequence perplexity. So there's something really interesting about scaling sequence data. How do we deal with the very different amounts of sequence structure and annotation data available? We ultimately we ran some experiments, but it is hard to run experiments at sufficient scale to make like really informed decisions about this. So essentially we ran some preliminary experiments on data mixtures for how much to weight different data sets, and ultimately settled on like roughly equal weighting of sequence only and sequence structure data sets. And then just incorporate function annotations where present and where available. Why do deeper model architectures yield better results from data? Yeah, I talked about this a little bit. I think having smaller vocab sizes emits like, or results in or it's better when the vocab size on data is smaller, then you tend to have better performance with deeper architectures. So for example, like this is true of images where you're representing like a pixel, which has, a tuple of RGB information, and relative to like the amount of vocabulary that's available for a language model, image models tend to like deeper architectures better as well. So that's one possibility. How do you construct val test sets that are not leaked at the same time? We mostly use temporal holdouts, which I think are easy to make, but which are also not exactly ideal because they can still leak information. There are some of our like internal validations, we did purge the sequences and from the rest of our training set using MMSeqs. And so we like actually just remove sequences from the training data that are too close to the validation set. What is the performance on the viral model? I don't know that we've benchmarked structure prediction on viral sequences specifically, so I don't know if I can answer that. It does tend to be better than the, anecdotally, it is much better than the open source model. Also, we fixed a bug in the open source model recently that may have led to worse structure prediction performance. So it is possible that it's worth running any structure prediction results that you had. Um, ASM3 able to reason about functions for uncharacterized proteins or detect remote homology. This is not something that we've particularly looked into, so I think you'd have to run those experiments yourself and see what happens. I would hope so. Was the variable masking rate purely for generation, or does it help with training stability? The variable masking rate is interesting. I think I mean, the primary reason was for generation, but it does seem to help, both in terms of like even in representation learning, it seems to help, but it also seems to have good training stability. I'm sure you could train a 15% mass language model though, with reasonable training stability. We do find that if you are going to go that route, as you scale the model, it's worth increasing the masking rate, or the average masking rate. So while at like 650 million parameters, like 15% masking is optimal. Once you hit 7 billion parameters, actually like 30% masking becomes more optimal. Long-term vision for protein representation learnings with LLMs. Yeah, there's a few things that we're interested in doing here, but I think that my ultimate vision for this is that there's a lot of information available about proteins in a very diverse set of data and formats. So this could be things like genomic context for proteins. So where does the protein sit in the genome? What are adjacent proteins to that in the genome? This could be protein ligand interactions and known interactions in that. But there's also, there's text about a protein. Like how does a human understand and reason about a protein? Oftentimes we're going to go and predict we're going to go read a paper and try and say and understand how and try and look at what experiments have been run on that protein, what have people looked at for it, and how can we and that those papers, those textbooks contain information that is clearly possible to learn from. And given the way that language models are moving in NLP, I think that there's something very interesting in trying to join those types of data together and create a model that can reason about biology, both in the way that like AlphaFold and ASM reason about biology, but also in the way that a human would reason about biology. Why unconditionally generated sequences are highly foldable, which sequences are not foldable by current folding tools like AF2 or ESM fold. I'm not sure I would say, well, one, the model's probably biased to producing sequences that it understands, and if the model understands the sequence, it's likely to be foldable. So that's one possible answer. It's also not that foldable necessarily. You definitely see significant improvements in both PLDT and PTM, although the improvements are much larger in PTM when you align the model to specifically produce higher PTM sequences. Is immunogenicity something you've looked at in function? Yeah, we haven't looked at immunogenicity yet. It's obviously something that we're very interested in looking at. Okay, I think this might have to be some of the last questions because I need to log off soon, but I'll answer a few more. Would you suggest fine-tuning all the modules for function annotation, or is it better to freeze the encoder decoder? I think it depends on how much data you have. If you use LoRa, I mean, I think you'd have to look at the literature. There's varying literature on how much you should use for LoRa, and what you should do for and how many parameters you should fine tune, and it's going to depend on your data set size ultimately. The license, I think the license, if you have questions about the license, you can email us or send it to or message us in some way. Yeah.
Awesome. Well, I'm going to actually cut it there for today to be cognizant of our speaker's time. So thank you so much again, Roshan, for being here and talking through ESM3 with us. If folks in the community still have questions, please feel free to email him or find a way to chat offline, or forward us some questions and we can get those to him. So thank you so much again.
Awesome. Thank you guys so much for having me.